I'm Jennifer Jones. I'm from the National Cancer Institute Center for Cancer Research. Our research for the ERCC2 is a joint project between Baylor, Harvard, and NCI. We're going to be looking at ways to study the cargo, the combined packages of information in extracellular vesicles. The overall goal is figuring out how we can read the information in these packets of information. The packets are these vesicles that are released by all of our cells, and the goal is to be able to monitor and survey the repertoires and populations of these vesicles in health and in disease. And so the purpose of this ERCC project that we're doing together is to be able to better read the messages on the inside and also on the outside of these packages of information of the vesicles to be able to understand how the cells are doing in the body. I first got interested in extracellular vesicles a bit by accident. It's one of those examples of science being a fun way to connect the dots. I was working in Susan Knox's lab at Stanford studying changes in the immune system in response to radiation. Found that one of the changes in splenocytes was upregulation of CD81, which is a molecule that another colleague of ours, Shoshana Levy, had cloned. And CD81 is a common marker on extracellular vesicles. And so that led me down the path of realizing that we could use these vesicles to study cellular responses and the state of the cells. And hopefully the goal is to be able to read the cargo to be able to monitor responses to treatment. I think this research consortium is very important, especially at this time in the field when we're studying extracellular vesicles and these other cargo carriers that carry RNA in the circulation. We're very much at the edge of the frontiers of what we can see and what we can understand. And so we learn a lot from each other. We help each other validate what we're seeing, and we learn to see things in new ways. And this, all in all, moves the whole field forward. I hope that we'll develop simpler, more widely usable methods for being able to detect, to study, to analyze, and to understand the cargo and the compositions of these vesicles in a way that will help us be better able to monitor disease and responses to treatment.